And now I have the great honor of introducing two of our graduate students who are recipients of the Ann Miller Gonzalez Grant. And our first speaker is Xinyi Yan. And Xinyi studies ecology, evolution, and behavior here at UT Austin. She's co-advised by Doctors Carolyn Ferrier and Amy Wolf. Her research focuses on interactions between plants and soil microbes. NIPSOT are big soil people, so they're going to be excited to hear about this. In a changing world, and how these interactions shape plant coexistence and community assembly. To answer these questions, she uses a variety of approaches, such as meta-analysis, theory, field experiment, and soil sequencing. Results from the project will help elucidate the impact of biodiversity loss and environmental change on plant microbe interactions and provide insights on native plant conservation and soil restoration. Thank you. Thank you.
Thank you so much for the nice introduction, and thank you everyone for coming here to see my talk. Yes, there it is. Yeah, as Laura mentioned, that I use a variety of methods, and today I want to just share the results, the preliminary results from a field experiment I'm running to study the effect of to study the effect of plant diversity and water availability on the soil fungal communities. So here's a picture that I took 2 years ago over here, right over here in May. And you can see the amazing wildflower blooming over here. So while we're appreciating the biodiversity, we may wonder, like, what does the current biodiversity loss and global change may impact on this biodiversity? So especially for Texas, we are actually facing very like a lot of change in the precipitation. So we're seeing more and more droughts these years, and it's becoming more and more extreme. So how does that paired with the loss of the native plants, which may be caused by invasive species, the change of land use, or being reinforced by those global change factors? How do those things all together shape the plant communities? And how does have cascading effects on other communities, such as the pollinator communities or the soil microbial communities, which is what I'm going to focus on today. So specifically, I study the interaction between plants and soil microbial communities. So we run experiments where we manipulate like factors to study the effect of biodiversity loss and climate change. Specifically, we manipulate native plant richness and native plant phylogenetic diversity. So for example, for those two, by comparing those two factors, we can have a sense of what are the different consequences of different modes of biodiversity loss. For example, if we can have plant species lost at random, so that's a decrease in the plant richness, the number per se, or we can have more closely related plants being lost together, more likely. So that's a decrease in the plant phylogenetic diversity. So all those two things can interact with the change in water availability or drought to affect our soil microbial community, i.e. the soil fungal diversity and composition. And we also expect that the subsequent shift in the soil fungal communities may have a feedback on the plant productivity as well. So to study all of this, we set up a field experiment at University of Texas Brackenridge Field Laboratory. So this is Austin. This is a map of Austin. So we're there right here. And this is the University of Texas campus. Here, if you can see it, the slim of green that's a little bit darker than nearby, it's our field station. So we have a lot of natural areas, but we also have experimental plots over there. So if we zoom in on that place I highlighted, this is how our experimental plots looks like. So note the scale bar. It's a very big experiment. And each individual square you see on the left is actually a 2 meter by 2 meter plots. Yeah, and then we have 144 plots, which I will tell you what's those composed of. So, and on the right, it's a picture I took in end of April, and you see different plants are growing there. So before I jump into how we arrange different treatments, I want to highlight the 12 local species we're using. They are all Texas native species. So we have 3 grasses from the Poaceae family. We have 2 nitrogen fixer from the Fabaceae family, and here's our beloved bluebonnet. And we have a Phacelia. We have 3 from the daisy family, from the family of Asteraceae. 2 mints, and a standing cypress. So those 12 species covers a range of growth forms as well as yeah, but they're all annual species. So we grow those 12 species each by themselves. So like in one plot, we just grow one species. So those are the monocultures. We also arrange those 12 species in different levels of richness. So we can grow them in 2 species, 4 species, 6 species, or all of them together. So within each of those like polycultures, 2, 4, 6 species, we also arrange those species in different levels of phylogenetic diversity. For example, let's say a 2 species plot. Those 2 species could be closely related, i.e. coming from the same genus, or they could be very distantly related. For example, a grass and a legume. Yeah, and it's not end yet. So we have so many different treatments. So we have those growing in the ambient plot, i.e. they do not receive any additional water. We also have the same exact species composition in water plots. So those water plots will receive like 100% more than natural water. And those are the 2 by 2 species plots, but within each plot, we have like subplots to examine the effects of herbivory on the plant communities. So we have like half of them being the open plot, a quarter of them having the full net. Remind yourself of the picture I showed with the net over it, that exclude all the herbivores. And we also have a quarter of the plot with like nets that didn't reach to the ground to control for shading. So all of them, we have all of those in 3 replicates in like 3 blocks. So we have 144 plots and 432 subplots total. It's a very big experiment. And actually, it's a very collaborative experiment. I'm only part of it. So it will be a lot of work to study the effect of all of them together on the soil community. But I'm eventually going to do that. But for here, I want to share the results of a subset of soil samples that I sequenced for pilot study. In fact, I collected all the 432 plots from 2 of the years, and I sequenced the 54 of them. So all of those, we have the monoculture of 3 species. We have the 2 species plots arranging low, medium, high phylogenetic levels, and we have all the 12 species plots. Those are all in the ambient treatments. But for the polycultures, the 2 and 12 species plots, we also sequence those in the watering treatments. So those are all in the open projects. No, like they receive herbivory, they get pollinators, but also we have those sequenced the soil from the 12 species plots with the insect exclusion treatment. So that's like gives each of them again in 3 replicates gives us 54 plots or samples in total. So I want to use this setup to help us answer several questions. The first one being, how does the plant richness, phylogenetic diversity, and water availability individually or interactively shape the soil fungal diversity and composition? So I'm going to directly jump into the results, but if you, at the end, I also have a slide prepared for what type of sequencing method or statistical analysis method used. So if you have any questions, feel free to save that for later. Okay, so for jumping to the first result, no single factor, no experimental treatment will explain fungal diversity or composition. So just a reminder, the plant richness, PD, watering, and insect exclusion are our 4 experimental treatments. So we'll have 2 rows of figures, but I'm going to walk you one by one. So on the top row, we look at how does each of the treatments affect the soil fungal diversity. So that's on the y-axis. So for example, for plant richness, the 2 levels of plant richness, the 2 species or the 12 species, for those 2 treatments, the fungal diversity looks pretty similar, right? It's like side by side next to each other. And statistical analysis also show that they do not differ from each other significantly. So on the next row, we want to assess the effect of those 4 treatments on the soil fungal composition. So the soil fungal composition, because there's so many different fungi in the soil, it's a very high dimensional data. But for visualization, we just sort of condense it into a 2-dimensional space where the fungal composition differs the most. But even for those most distinct 2 dimensions, the fungal composition do not differ from each other in the 2 species plot and 12 species plot, indicated by the 2 colors. And you can see the dots. It's like those are like samples in each of the plots are like well mixed. You cannot tell them from each other. So that's a way to read this, the bottom row. And if we run the statistical analysis, it's like we can say that plant richness does not significantly influence soil fungal communities composition either. So if we look into all the other treatments, we can see that for fungal diversity, different levels of plant PD, watering availability, and insect exclusion do not really significantly change or shape the soil fungal diversity. And similarly, they do not significantly give rise to different distinct fungal composition either. So are we saying that, so it seems that the soil fungal community is so robust, they don't really care what's going on in the world. but no, that's not what I want to tell you. So we follow by our second result, which is actually we find an interesting interaction between water availability and plant phylogenetic diversity on shaping the fungal diversity. In another word, like how plant diversity loss influence the soil fungal diversity really depends on the amount of water those plots receives. So let me show you some graph, but I will walk you through it. So on the x-axis, we have the numerical values of plant phylogenetic diversity. So those are calculated by, so we construct the phylogenetic tree of those plants, and we calculate the branch length between any 2 species. So anyway, lower values means that those 2 plants are closely related to each other. Larger values means they're distantly related to each other, and meaning that they're like more phylogenetically diverse. So on the y-axis, we have the fungal diversity, and we have our 2 water treatment over here. So let's look at the red one first. So that's our ambient plot, and all the dots showing individual samples for those ambient plots. So we can see that in ambient environment, fungal diversity really decreases with plant phylogenetic diversity. Whereas if we look at the blue line and blue dots, it's the opposite. Fungal diversity increases with plant phylogenetic diversity, i.e. it's like a diversity begets diversity scenario. It only happens with the addition of water. Yeah, so this one, I mentioned the y-axis is the numerical value of the phylogenetic diversity. So we actually calculate that first, and we categorize into low, medium, high. So if we use that, we can see it's a similar trend. And there's like, but however, the statistically the interaction is not as significant. Okay, so that's our first question. And we find the results that our native plants diversity sort of really interacts with the effects of global change on the soil on the below ground world. But we also are interested in if the shifts in the below ground world can feed back into the plant community, i.e. can they change the plant productivities across different treatments. So note over here, we have a double arrow because on the left, the plant richness, PD, and water availability are all experimental treatments. So we can say, we can conclude the causal effect of those on the fungal diversity. But the fungal diversity and the plant productivity are both just measured. So we measure them at the end of our experiment. So we can only infer association, like rather than causation. So that's why the double arrow. So the result is that we also find another interactive effect of fungal diversity and water availability on plant productivity. So again, we have our first plot. Right now, the fungal diversity instead on the y-axis, it's on the x-axis. And we want to see their relationship with the y-axis, which is the total plant percent covered. We use that as a proxy for productivity. So it's like how much of the plant covers the ground, basically. The more it is, the more productive the system is. So we can see that there's also like a cross of those two different colors. That's an indication of the interaction effect. So again, we have the red as our ambient plot. In those plots, we should find the negative association or negative correlation between fungal diversity and plant productivity. Whereas in the water plots, we find that there's a positive relationship between those two. So over here, I use total percent cover as an indicator of productivity. What if we just use plant biomass? So again, it's a pretty similar trend, and so the statistical test shows that it's slightly less significant. So I know this is a lot, but I'm going to put in everything together and give you a take-home message at the end. So together, okay, this is a fun part. Stay tuned. So, but this plot, we have just seen it, so it's nothing new. Don't be afraid. So note the plus and minus sign. Okay, so the blue one means that in the water plots, the red one means in the ambient plots. Red, because in our ambient plot, it's actually a little bit ambient drought. That's why I use red. So we find that so this is our first result, and this is our second result. And we find that whether there's a positive or negative relationship, or like positive or plus or minus sign, depending on which treatment they have. So I will walk this through. Okay, let's focus on the blue plus signs. So we find that soil fungal diversity increased with plant phylogenetic diversity in water plots. Also in those water plots, the increase of the fungal diversity will further increase the plant productivity. So we will probably expect that in water plots, if we draw an arrow between plant productivity with plant diversity, there's going to be a positive relationship, right? Because all the routes are positive. And in the ambient plot, it's an opposite. We find that plant phylogenetic diversity decreased soil fungal communities, but those decrease in the soil fungal community will actually increase the plant's productivity. So how do we but we actually have the data to see the plant productivity, diversity productivity relationship, but it's not significant. So it's like, okay, and it doesn't really differ by which which water treatment they receive. I know, I was scratching my head, but so I have a little bit takeaway for this, but I have more ways to go, which I will talk to you after this. So we find that the interactive effect is very pervasive, and those really have interactive effect between global change factors. Specifically over here is the water availability and droughts, and native plant diversity loss of the soil fungal communities, as well as the soil fungal communities cascading effect back to the plant communities. However, while soil fungi are key players, additional process must be considered to fully understand the diversity productivity relationship in the plant community. So yes, our the overall is like we know that things are complex, but there are plenty of evidence showing that there are feedbacks between our native plants communities and the soil communities, and their relationship really depends on the water availability. However, one caveat is that I mentioned this is only from a subset of those samples. But I actually collected all the soil samples from all of the treatments from both 2002 and 2003. So my way forward is to sequence all of those like 288 samples from 2 years. So actually over here, I only very briefly mentioned the herbivory treatment, but because of lack of funding, I don't have money to sequence like 864 samples. So I'll wait for that for a little bit. But for now, I can really study the effect of plant richness, plant phylogenetic diversity, water availability, and year, their effect on the fungal community. And note that most of us live here in Texas, and some of us live in Austin. We know that different years have different levels of water availability. So that's another way to study global change factors effect of fungal communities as well. So right now, I'm actually preparing all the 288 samples. So stay tuned. I'm very excited. So at the very end, I want to really acknowledge my collaborators and anyone who has helped me with the experiment and the lab work. I also want to acknowledge my funding sources. And especially I want to highlight the Native Plant Society of Texas and the Gonzalez Graduate Research Award, because with that, I sequenced all my my 54 soil samples using that grant. It's a very expensive process. I could not have done that without it. Yeah, thank you so much. And thank you everyone for listening. Oh yeah, we already have, sorry. We have time for maybe one question. Oh, okay, we have time for one question. Oh, that's a great question. So actually, this experiment was originally designed by 2 graduate students a year before me. So I sort of hop onto that, but I know the process. So so we have one Texas native. Sorry, we have one of my lab mates grew up in Texas. So she knows what species grows well. And basically, because we have experiment, we want all of our experimental species to grow. So that's one way. And we also want to make sure we cover different groups. We want to have some grasses, we want to have some nitrogen fixers, we want to have some asters and other species. So that's sort of the 2 criteria that we chose those 12 species, and how to arrange them in 2, 4, 6 species, diversity and richness levels, is that we, like especially for the phylogenetic diversity level, we calculate the branch length of between any of the 2 or 4 or 6 species on the phylogenetic tree, and we group the ones that's really closely related to each other in the 2 species plots, the ones that's sort of in the middle for the median phylogenetic diversity plots, and the ones very distant related for the high diversity plots. And note that we have like 3 blocks for 3 replicates. Those phylogenetic diversity treatments are actually having different plant composition. So that we can distill the effects of specific plant composition from the pure diversity level, pure effects of diversity levels. Yeah, and if you have more questions, you can email me. So yeah, I'm happy to talk more. Yeah, thank you so much.